Sir, good morning, sir. Start பண்ணிடலாமாங்க sir?
Yes, shall we start?
Yes, sir. Yes, sir.
Yes.
I'll just give you a small introduction, sir.
Okay.
Very good morning to all the participants today. We are having our KA's 12th online webinar. I welcome all of you for this webinar. The webinar is on role of soil microorganisms for sustainable agriculture. I invite our newly joined principal, respected Dr. B.J. Pandian, sir, to give a brief intro on this webinar. Sir, please, sir.
Yeah, okay. Thank you, Inay Kumar. Dear guest speaker, Dr. Panneerselvam from the esteemed National University of Rice from Cuttack. Dr. Inay Kumar, seminar organizer and assistant professor microbiology. Faculty members of KAA. My dear students of second year, third year students of the KAA campus. And also other participants who are attending this webinar from the outside the campus for the enrichment of the knowledge. At the outset, on behalf of KA, staff, faculty and management, I extend a warm welcome to our guest speaker, Dr. Panneerselvam, who is working on this microbiology in one of the leading institutes in India, that is National Rice Research Center from Cuttack. And also I extend warm welcome to all the students, friends who are very eagerly attending the seminar as a part of this curriculum and through webinar. And also I extend all the participants from the outside also for this seminar on behalf of the staff and management of the KA. So as far as this seminar is concerned, everybody knows that our Indian agriculture is facing several crises. And one of the important issues in Indian agriculture is the land degradation, or we can say it is poor soil fertility or environmental degradation is one of the important issue which causing concern to everybody in the Indian agriculture. So over the decades, application of chemical fertilizers, which resulted in the stagnation of the yield and poor soil fertility status also. So lack of inclusion of these biological organism, which causes concern to everybody. So today Indian agriculture is making a new dimension and bringing this biological system into the agriculture, and an hybridization of bio inputs along with limited chemical inputs definitely will play a major role as far as sustaining this Indian agriculture and getting the more yield also. So under this context, the soil microorganism plays a very important role. So soil microbiology is one of the oldest subject. Everybody knows the Tamil Nadu is the leading in most of the soil microbiology, and I'm happy guest speaker also from Tamil Nadu, because most renowned scientists, Dr. Rangasamy, most of the Dr. Kannayan, and most of the renowned scientists are from Tamil Nadu, who has worked even 1960s also, from the 1960 and for different decades also. So Tamil Nadu has got very good strength in the soil microbiology, and both in the delivering the subjects and developing the technologies also. So, and this contrast, organizing such a webinar is a very useful one also. I am happy. I joined only 1 week back. Really, I'm happy to inaugurate though it is a 12th seminar for KA, for me it's the first seminar. So it is we started with the microbes. Maybe we ended with the elephant also. A bigger thing also, from the nanoparticles to bigger particles. Though it is a very microbes, it is a very essential thing. So which for the doing catalytic activities and bringing the nutrients also. So in this context, organizing such seminar on soil microbiology as a part of curriculum of AGN 301 agriculture microbiology. So role of soil microbiology on sustainable agriculture, and listening this outcome from the guest lecture of this National Institute is very important. I really am happy and I thank Dr. Inay Kumar bringing such a stalwarts from this National Institute, introducing to our students. So we as faculty, many times visited Cuttack, we know Dr. Panneerselvam and other directors also. But most of the students should be familiarized and by learning the lectures. I hope all the students should make a keen attention, and such type of lectures will be very useful for your curriculum and also knowing the subject in a better manner or in a comprehensive way also. So I hope I am introduced this topic very nicely. I think once again I welcome our guest speaker, Dr. Panneerselvam sir, to deliver the lecture on this important topic. So I will hand over to mic to Dr. Inay Kumar.
Thank you very much, sir, for your nice introduction and for your advice to the students for their keen interest. Thank you very much, sir. Now I introduce Dr. P. Panneerselvam. Dr. P. Panneerselvam is one of the leading agricultural microbiologists in India. We are proud that he was our alumni of TNAU. He was our senior when we were doing masters, and he has done a lot of work in agricultural microbiology. So he has developed several technologies. He has transformed the science of agricultural microbiology into several technologies. One of few technologies I list out here because his resume goes along more than 10 pages. It will take me 1 hour to read all the important achievements. But I would like to highlight here his important technologies in microbiology. So he has developed a microbial consortium when he was working in IIHR, that is named as Microbial Consortium, along with his colleague. And this biofertilizer alone has been licensed to 33 entrepreneurs for commercialization. And he has received several awards for his best work in soil microbiology, and he has developed soil-less cultures for WAM multiplication. He has also developed Actino Plus for soil enhancement. He has been awarded with so many awards, but due to want of time, we should hear from the guest speaker. I invite the respected Panneerselvam sir to give his talk. Thank you very much, sir. Please, sir.
Thank you. Thank you, Inay Kumar. First of all, everyone, student friends from Kumaraguru Institute of Agriculture. First of all, I thank management, principals, and my good friend Inay Kumar for this great opportunity to share my little knowledge in the field of microbiology to you great people. Is it visible, Inay?
Yes, sir. Yes, sir. The slides are visible. You can just put it in screen. Yes, sir. Yes, sir. It is right. You are
Once
good.
again, I thank Kumaraguru Institute of Technology for given this great opportunity to share my knowledge in the field of microbiology, particularly for the role of microbes for sustainable agricultural productions. There is no doubt we have soil, air, water, of which the soil is a major source for these microbial resources, wherein we are growing plant and microbes are being associated with this soil. Soil is a major source, no doubt at all. But the thing is, once if we understand the proper interactions between the soil, plant, microbes, so definitely we can make magic in agricultural production. There is no doubt at all. The microbes play major role in our agricultural productions, particularly sustainable agricultural productions. But in India, populations are increasing day by day, not only in India, everywhere. But in one side, population is increases. In other side, industrializations urbanization is also taking place. So wherein we have great pressure to increase agricultural production per unit area. There is no doubt at all. When we want to increase the production, apart from water source, we must apply fertilizers, we must apply pesticides for enhancing agricultural productions. But the indiscriminate usage of these inorganic fertilizer, pesticides is severely affecting the soil health, particularly these microbial diversities, microbial properties in soil. But maintaining soil health is very important to sustain agricultural production. Then how best we can reduce this usage of inorganic fertilizers and fertilizers? There are many ways are there, of which the research documents as well as the commercial available technology also well proved that by explorations of beneficial microbes from soils, so definitely we can sustain agricultural productions. We can considerably reduce the amount of use of inorganic fertilizers and pesticides. So before going to discuss this main topic, first we have to conserve that water, reduce the usage of inorganic fertilizer or synthetic fertilizer, pesticides. Same time, we have to promote the biodiversity in crops grown and ecosystems. Above and all, the sustainable agriculture also should focus maintaining the economic stability of the farm. Same time, it should improve the quality of the farmer life. If we made everything, so this is called as sustainable agriculture. But today, our talk is how these microbes will meet or how these microbes will support its own area, wherein we are regularly using fertilizers, pesticides for increasing our food productions. No doubt at all. After green revolutions, we have increased our food grain productions, even we can say more than 5 to 5.5 times. If you see this data, based on this 2018-19, we have used nearly 27 million tons microbial cultures, or by using these microbial resources, we can considerably reduce this inorganic use of fertilizers. What we also have proved that we can save nearly 20 to 25%. Even if you keep like lower percent, even 15, if we save use of inorganic fertilizers by using this microbial technology, so definitely we can cut short for 3 to 4 billion tons usage of inorganic fertilizers in our India. If you see this biofertilizer production data in our country, here I have taken last 1 decade data. You see 2012-13, so we have produced nearly 45,000 tons of this biofertilizer in our country. But what report says, even if we use 25 to 30% area in our for biofertilizer usage in our country, we need around 5.5 lakhs metric tons of biofertilizer in our countries. But you see these 5 years back data, 2014 and 15, we have produced only 80,000 metric ton. Even if you take 1 lakh tons also, we have a lot of scope to use these biofertilizers in India. If this biofertilizer technology or this microbial inoculant technology reach each and every doorstep of farming communities, so definitely we can reduce use of these inorganic. Soil has millions of microbes, no doubt at all, but most of the microbes are uncultured in nature. 5 to 10% we can culture. Even this, by culturing this, if we see in our India as well as in other countries, so these are, commercial bioinoculants available in our country. For example, nitrogen fixing biofertilizer. Yes, this is one of the important biofertilizers. In India, if we see 1920 onwards, people started using rhizobium. But 1960 onwards, people started using rhizobium more in our leguminous plants. Apart from this nitrogen fixing biofertilizer, we also have phosphate solubilizing biofertilizers, both bacteria and fungi. And another important microbial agent, that's arbuscular mycorrhizal fungi. This is endophytic beneficial mycorrhizal fungi being associated in crop plants, wherein apart from mobilize the nutrients away from this root region to the plants, it also play a major role in biotic abiotic stress management, ectomycorrhiza. Wherever we go in forest region, we could see some mushroom-like kind of fungi beneath the tree plants. It is called ectomycorrhiza. But some plants must some mycorrhizal association. For example, orchid, it must need mycorrhizal association for their better growth. Apart from this nutrient management, we also have plant growth promoting rhizobacteria. In some places, people use it to sell like biostimulants. Other than this major, the nutrient meeting biofertilizer, we also have silicate zinc solubilizing biofertilizers. Here you can see these pictures indicates that how we can improve the soil health or plant health by exploring these microbial associations. First, we will start from this one, the nutrient cycle. Yes, we have 141 million hectares agricultural crop land, wherein we are producing the huge amount of agricultural biomass. If we properly recycle this agricultural biomass produced in our countries, so we can reduce usage of these inorganic fertilizers also. By recycling effectively this organic recycle waste, there are many microbes available in soil, and also same time we have to improve. Many decomposer, including TNAU, Indian Agricultural IARI, Delhi, we also have developed some product. If we use effectively those microbial cultures, so we can convert this waste into valuable manure. Another thing is the plant protections. The plant protections also very important aspects is, because of ravage of many pests and diseases in our agricultural crops, reducing this yield in most of the agricultural plants. Wherein, if we properly utilize these microbial agents, so we can control this pest and disease, through which we can reduce pesticide usage in our agricultural systems. The nutrient management, it's no doubt at all, everyone knows how these microbes fix the nitrogen from atmospheres and convert those nitrogens into ammonical form to the plant systems, by which also we can save considerable amount of nitrogen. In soil, some of the microbes will produce some polysaccharides. Those polysaccharides are very important for soil aggregation process. That will improve these water holding capacities of the soil. Apart from this, some of the nutrients may be available in soil, but may not available to the plant systems because some of the applied nutrients will be get fixed into the soils, so plant may not take. Wherein this microbial associations is very essentially required by releasing certain kind of organic acids, enzyme. These microbes will release those nutrients, then plant will easily will take those nutrients. When we apply some minerals like rock phosphates kind of minerals, even by which also help of these microbes, we can convert those minerals into valuable fertilizer for these plants. Other than these nutrient cycling, plant protections, the PGP activities, microbes play a major role in plant growth promoting activities. For every crop, for their growth, needs some phytohormones, IAA, GA, kind of substance is essentially needed to improve their plant growth. Most of the microbes are well documented and proved that play major role in secreting those compounds through which it increases this root growth in these agricultural systems. So overall, the combinations of all these factors will improve the plant nutrient uptake and plant disease and pest and management, which in turn increase this productions in our agricultural system. So here, the microbes, play in our plant systems in two different manners. One is kind of, second one is kind of indirect mechanism. If we see direct mechanism, microbes directly will involve, for example, it will degrade this organic waste, directly will supply this, nutrient to the plant systems. Some of the microbes, as I already told, directly will produce these indolactic acids, gibberellic acids, through which it will induce the growth. Some of the microbes directly will be involved in phosphate solubilizations or nutrient solubilization, potash or zinc, even manganese solubilization. Directly it will helpful to the plants. But some of the microbes will indirectly help the plant system. For example, not only in plant system, in our human bodies also, we need immunity. For that immunity, to improve this immunity system in the plants, these microbial associations are very much important. Some of the microbes will trigger some metabolic pathway in plant system that will induce some immunity system in the plants, through which we can make the plant resistant to many pests and diseases. Apart from that, indirectly it will kill some, harmful pathogens by producing some metabolites, HCN, some enzyme. For example, here I have mentioned siderophore productions. Yes, that is one of the important compounds through which it manages the diseases. For example, by producing these siderophores, the microbes will chelate this iron compound. This is very much essential for most of the organisms for their metabolic function, through which it will suppress these pathogenic organisms. So like that, in many ways, the microbes are play major role in crop production. Here I have put one more slide. Whenever we select these microbial cultures, we have to select that organism should have some multiple effects, multiple beneficial effects. For example, here I have highlighted the phosphate solubilizing bacteria. The same phosphate solubilizing bacteria Apart from phosphate solubilizations, it also has phytohormone production. It also has disease controlling mechanisms. It also has some antifungal metabolite production, nitrogen fixations, and other kind of things. So that's why whenever we want to select any microbes for agricultural purpose, instead of targeting some specific functions, if that organism has some multiple functions, then performance will be better. So another important thing is rhizosphere competence. Rhizosphere competence is one of the important criteria for selections of the microbial cultures to develop inoculant preparations. For example, some of the bacteria, whether it may be fungi, bacteria, or actinobacteria, perform better in laboratory conditions. When we introduce into the soils, it may not work. So that's why the rhizosphere competence is very important. So before selections of organisms, we should keep this into mind. Yeah, another important. Actually, this biofertilizer usage started in India, I told 1920 onwards, but 1960 onwards, people started using this biofertilizer in most of the states. But those time, even now also, the people is used to give special attention to use single organism. But now the new concept has come, that is called microbiome. Similar to our humans like gut microbiome. By improving gut microbiome, we used to induce immunity system. Similarly, in soil, instead of encouraging single microflora, so we try to improve that microbiome in these rhizospheres or spermospheres regions in soil. So through which we can get better result. So this is kind of recent concept is going on in most of the world. No doubt at all. So already many research papers have well proved that by improving this microbiome in our soils, we can, manage pest and diseases. Apart from pest and diseases, nutrient management, which in turn will increase our agricultural productions. So that's why now people are giving much importance. Instead of using single strains, they are focusing on this microbial consortium. Now plant breeders also having some collaborations with the microbiologists. They want to produce, they want to breed the plant which is highly responsive to the microbes. Particularly in our Rice Research Institute also, we are working on how best we can, breed for mycorrhizal responsive variety to produce the plant for resistant for the phosphorus and nitrogen. So that's why to apply these millions of microbes or maintaining millions of microbes in soil is very difficult. So that's why now most of the groups working on identifying some keystone microflora. One simple example just I would like to highlight. Suppose in just one village, in village we have for 5,000 people, wherein we want to control those 5,000 people, or if we want to convince those 5,000 people, it's very difficult. But in same village, some of the important peoples may be there. By approaching those 2-3 peoples, we can control the entire village. Similarly, in soil, peoples are focusing to identify some keystone microflora. By identifying the keystone microflora, if we make some product, if we introduce, that microflora will maintain the microbiome in rhizospheres or spermospheres or in soil, through which we can alter this microbiome, so by which we can manage these pests and diseases and nutrients. But here, I already told this climate change also plays major role in changing microbial diversity in our agricultural soils. Here we have done one research in our Rice Research Institute. Now people are focusing that the carbon dioxide concentrations increases day by day. They are predicting that in 2050, the concentrations may reach 550 ppm. They are predicting in 2100, it may go even 700 ppm. Suppose if we increase this carbon dioxide concentrations, what will happen in this microflora? This is very interesting things. As I already told, this mycorrhiza is one of the important biofertilizers, is commonly being used in most of the crop plants. If you see in India, most of these bioinoculants, particularly this mycorrhiza, is prepared from by using whether Clardioglommus or glomus. Most of the products containing only this genus. But what we observe, if we increase this carbon dioxide concentration for a long time, this particularly population, this Clardioglommus glomus, has drastically reduced. Even we can say that 90% of associations is reduced. Whereas the other group like Scutellospora, Apellospora, those genus are increased. Okay, what I want to convey message from this slide. So depends on the environmental conditions and soil. We have to identify what is the dominant flora, wherein we have to select the efficient one. By using that efficient culture, we have to introduce or prepare these microbial products. In continuation of our earlier studies, we also have proved that you all know that rice is being cultivated, particularly in wetland conditions. In water stagnated places, we are growing rice. But most of these beneficial organisms in our country are aerobic organisms. There is a school of thought. When we introduce this mycorrhiza in wetland soil, mycorrhiza may not work. This is the their thinking. So to understand this concept, we have done some systematic studies. Here we have proved that if we use the organism capable to survive in this wetland, that means in native microflora, definitely we can increase the grain yield. Apart from increasing grain yield, we could save use of phosphorus fertilizers, particularly in rice. Similarly, now the peoples are using only fertilizer. In some places, they will not use any organic manure because their main task is to increase their production. To understand these concepts, we have conducted one more studies. Even my colleague has conducted very nice studies. If we use continuously this inorganic fertilizer, particularly this nitrogen, urea, to our agricultural productions, particularly in rice cultivations, what will happen in these microbial diversities? He has proved that if we use only this nitrogen application to our soil, this will drastically reduce some nitrogen fixing bacteria and cyanobacteria in our soil ecosystems. So we always should not depend on our already proved new microflora, because soil is have millions of microbes, wherein always we should find out some new species for our targeted interest. Here, for this rice leaf folder and stem border management, we have been using this BT. It is called Bacillus thuringiensis. It's one of the important biocontrol agent is being used for, insect management. But while searching in these microbial diversities in rice ecosystem, we could come across one new organism that is called Spermanella. So it has, tremendous biocontrol potential against this rice leafholder and stem borer, equivalent to these Bacillus thuringiensis. Here, what is the message? Instead of using our already proved cultures, always we try to find out some new species from soil, so by which definitely we can bring out major changes in our agricultural production. Similarly, we had one DBT project where, because Sikkim is an organic state, wherein we have been given task to develop the bioinoculant package for Sikkim, we come up with some formulations. So here you might have known this Bacillus amyloidecufficin subtilis, but we come across a new species, the Bacillus luciferensis. This organism has, tremendous biocontrol potential for managing leaf blight and leaf folder. So that's why always we try to find some new species from soil. Yeah, so far I have given the overall role of these microbes for our agricultural productions. I specifically emphasize we have to use, instead of using single cultures, we have to use the microbial combinations. Suppose if you are recommending some bioinoculants to the farmers for nitrogen one product, phosphorus, potash one product, zinc separate product, for biocontrol agent separate product. So like that, if you recommend to the farming community, sometimes farmers find some difficulties by all these products, apart from these cost involvement. Sometimes farmers may miss to use any one of these products. That's why we have to develop some simple product which should be, acceptable, adaptable by the farming community. Instead of using these single strains, if we use these combinations of many strains in single carriers, we can get better result. So those things, based on our personal experience, I would like to share those informations with you people. Yes, when I was in Indian Institute of Horticultural Research, actually I should thank IAHR, ICAR. They given the better opportunity for us to develop these products. Actually, with my colleague, even Dr. Selva Kumar, with this team, so we developed 4 technology, the consortium-based technology. But the technology is, working well for this horticultural as well as agricultural crops. We have developed ARCA. ARCA means the product developed from IAHR. Like PUSA. PUSA means the product developed from IARI. Similarly, whatever products start with ARCA, maybe the product developed from IAHR. We developed ARCA Microbial Consortium, ARCA Actinobacteria, and soil-less AN from Medina. So quickly I will highlight by using these products, so how we changes in horticulture as well as agriculture. Why I have selected these 4 products? Because in soil, bacteria is there, fungi is there, mycorrhiza is there, and most of the product is either from bacteria or fungi or mycorrhiza. But here I have selected ARCA Microbial Consortium is bacteria-based product. ARCA Actinoplas is Actinomyces based product. So fermented cocoa bean is a different kind of technology. The product is enriched with some beneficial microbes. Then another one is mycorrhizal. This ARCA Microbial Consortium is a carrier-based consortium. This consortium contains nitrogen fixer, phosphate solubilizer, zinc potash solubilizer, and plant growth promoters. But the importance of this product is whatever organisms we have used in this product, all the species are new species. Only one example just I want to highlight, phosphorus. For phosphorus, we know it's a Bacillus megathelium. It's a well-known organism is being used as a phosphate solubilizing biofertilizer. But here we used the Bacillus aureopatai. Actually, this organism initially reported from stratosphere. They thought that these organisms will survive only in like stratospheres. Then later, one more scientist from soybean, they have reported these organisms from rhizospheres. But proudly we can say that by using those organisms, we come out with this product. Now that product is working across many crops. Here the concept is the farmers, if they use these products, they need not apply any biofertilizer related to nitrogen fixers, phosphate solubilizers, zinc or potash solubilizers, or plant growth promoter. That is the advantage of this product. Normally, we recommend 5 kg solid waste per acre, even 10 kg, even you can apply up to 15 kg. Also developed for these liquid cultures. These liquid cultures is very easy for application in this horticulture. When we use this Microbial consortium, we can improve our yearly germinations. For example, for tomato, we have to wait. We have to wait for 25 to 27 days to produce the seedling. But when we use this microbial consortium, we can reduce this nursery period 3 to 4 days, even 21 to 23 days, the seedling will be ready. Apart from reducing this nursery period, it will improve the seedling vigor. So overall, we have conducted many trials. Now this product also available with these farming communities. One thing I want to say, almost all this technology we have been commercialized to more than 30 to 35 private companies, including KVK and government institutes. Now this product is very popular in southern India, particularly in Karnataka. Now slowly is gaining importance in other parts of India. We have well proved that by using this microbial consortium, we can, apart from reducing this 25% of nitrogen, phosphorus, potash, we could increase 10 to 16% yield in different kind of crop plants. Here you can see the difference. Normally for horticultural crops, people will use this, decomposed cocoa peat as a base media to raise the seedling. The left side is controlled, right side is treated one. When we use this microbial consortium for the seedling productions, here you can see how the difference between the control and microbial consortium seed. We can, produce or increase the robust root growth by introducing these organisms in horticultural plants. So like that, we have carried out in many vegetable crops, including plantation crops, cauliflowers. Here you could see chili, brinjals, and other crops, how these microbial consortium interventions improve this seed growth, root growth. As I already told, we have conducted in many vegetable crops. By using this technology, so we can increase 10 to 16% yield. Apart from increasing yield, we can reduce 20 to 25% of usage of fertilizers in horticultural industries. So this is another view. Ginger, the ginger rot is very serious problem. Here our idea is, as I told earlier, still we don't know, we have not proved. But the people are saying that when we use this microbial consortium in our field, we are not getting any disease and other kind of things. So what I personally feel, as I already told, the keystone microflora. By introducing some important microflora, we can improve that microbiome in soil as well as rhizosphere. So like that, by using this microbial consortium in ginger, the peoples are managing this ginger rot problems in most of the places. So like that, we have conducted, many success stories in horticultural crops. After I joined NRRI, National Rice Research Institute, then we have evaluated how these products will work for rice. We evaluated these products in wetland as well as aerobic rice. We have evaluated these products for more than 3 seasons. So we come to know that when we use this ARCA microbial consortium, that I will tell later, that is ARCA Actino Plus. So we can, reducing this, apart from reducing these fertilizers, we can get yield at par with these 100% NPK applied treatments. This is in the case of wetland crop. Whereas in aerobic rice, we could increase yield 9 to 14% when you use this microbial consortium for these rice cultivation. So what it indicates, so this microbial consortium, apart from horticultural crops, it is working or performing well for other crops also. So after commercializations, now people started using for other agricultural crops also. Then frequently I used to get feedback from the farming community as well as scientists from ICAR. This product is working across many crops in our India. So this is another product, Arca Actino Plus. This is Streptomyces based consortium. Everyone knows these antibiotic productions in our India, in our countries, not only in our countries, in universe, 70 to 75% of the antibiotic is being produced by the Streptomyces. The Streptomyces is one of the dominant microflora next to these bacterial populations in our soil. But it created revolutions in industrial field. Then we thought that we can explore those Streptomyces species for our agricultural productions. Actually, this idea, how we got, in pomegranate, particularly in Karnataka, there this nodal blight, pomegranate wilt, is a serious problem. We used to meet some farming communities. They always used to say, "Sir, try to bring out some product against for this nodal blight management." Then we thought that, we asked, then what kind of strategies you are following for this nodal blight management? This disease caused by this Xanthomonas species. Then they told, we are using a lot of antibiotics to manage this disease. Then we thought that Actinomyces is one of the number one organisms is producing these antibiotics. So I can try these Streptomyces for inoculum preparations. With that, we have started. Then we have prepared. Then we have formulated this product, Arca Actino Plus. This is the combinations of 3 Streptomyces species. This technology officially released from IAHR, and also we have licensed to private entrepreneurs also. Similar to Arca Microbial Consortium, this product also has wonderful for plant health improvement and soil health improvement. We have proved in many horticultural crops. So finally, I can say that by using this technology, we can increase 13 to 20% yield. But one beautiful observations is this Arca Actino Plus as well as Arca Microbial Consortium has good compatibilities. That's why in most of the places, the peoples are using this streptomyces-based product along with this ARCA microbial consortium. Whenever we use these combinations, the peoples are getting better performance for this disease management apart from enhancement of yield in most of the crop plants. This is in tomatoes. We have proved this is one variety, good variety ARCA itself. Some of the farmers, by cultivating even tomato, they became pro. Here, what is the message I want to convey? They have proved that by using either ARCA microbial consortium or ARCA Actinobacteria or combinations of these products, if we use per plant, we can increase around 1 kg. On an average per hectare, this ARCA Rakshak plant may be 10,000 plants. 10,000 plant means each plant may produce 7 to 8 kg, in maximum cases 10 kg. If the targeted yield is 100 tons, even if you produce the 50 tons. By introducing this microbial technology, if you increase 5 to 6 tons, if you put like 5 rupees or 10 rupees per kg, at least the 50,000 additional income farmers will get by spending 1,000 to 1,500 rupees per hectare. So like that, we have conducted in many vegetable crops wherein we got very excellent result for enhancement of the apart from reducing inorganic fertilizers. So this is one interesting thing. The people, we have not developed exclusively for plantation crops. Then by seeing the performance of this product, some farmers came from some poor, particularly in Karnataka. They have started using because there in pepper, this wilt problem is very serious. So they started using this product. Now, even you can see through internet also how this product is working for this plantation crop to recover the plant health, particularly in pepper from this poor region and other parts of the countries also. So this is another one important technology, ARCA fermented cocoa bean. In my earlier slide, you might have seen the people earlier, they will use this decomposed cocoa coir waste for rooting media to produce this seedling. To get 1 ton, farmer has to spend nearly 3,000 to 4,000 rupees. By using this technology, we can reduce around 50% cost. That means instead of spending 4,000 rupees, so by spending only 2,000, 2,500 rupees, we can produce 1 ton is fermented. What is the beauty of this technology? As I already told, whatever technology we develop, that should be easily adaptable by the farming community. If we develop such kind of technology, we can make success among these farming communities. This is one of the technology example for one simple technology made great impact in horticultural industries. Here, our idea is not to decompose. Here, our idea is we want to find out some toxic compounds which hinder the plant growth. Those compounds, by introducing some microbial cultures, we break down. Within the month, these products will be ready. Whereas in normal cocoa peat preparations, we have to wait for 3 to 3.5 months. And we have to spend a lot of energy for washing, pasteurizations, and other kind of things. By using some microbial cultures, we developed some fungal consortium. Along with this ARCA microbial consortium, we could produce this fermented cocoa peat within month. This is the old methods people used to produce this decomposed coir waste, wherein the people will use lot of water. After washing, it will be subjected for pasteurizations. after pasteurization, they will introduce some cultures. It's a lot of energy, time-consuming process, and we have to wait for 3 to 4 months. But by adapting our technology, we could produce this fermented cocoa pit within month. So this is the Arca fermented cocoa pit. So by using this Arca fermented cocoa pit, if farmer raise the seedling will be colonized by this Arca microbial consortium. When it goes to the field, so automatically it will be, proliferated in field conditions. So this is some success stories I want to say. Actually, I should thank these persons. Because of his encouragement, we could develop so many technologies. For example, every day we use it to spoil even 500, 300 to 400 seedling. But these farmers is used to encourage us. No, sir, we try. Definitely we can come up. Now, he started business with nearly 250, 300 rupees. Now he is a kind of, his turnover is more than crores. By adopting this technology, he is one of the number one entrepreneurs or nursery entrepreneurs in Bangalore. Yeah, so I already told now this mycorrhiza. Mycorrhiza is one of the endophytic beneficial fungi being associated with most of the crop plants, wherein this mycorrhiza will act as a root hairs or kind of roots will help the plants for nutrient management, biotic and abiotic, stress management in agricultural crops. When we use efficient this mycorrhiza in our agriculture, initially we thought that these organisms will mobilize only phosphorus. Now they have proved that these organisms also support to provide nitrogen, potash, calcium, magnesium, zinc, and other micronutrients. Apart from this, these organisms also play a major role in bringing resistance or immunities in the plant systems against disease and drought. For example, drought means a root sometimes cannot enter inside capillary pore, but this mycorrhiza will enter inside the capillary pore. It will take the water. And also these organisms also producing a kind of protein that is called soil aggregating proteins. By apart from this mycorrhizal network, also by producing this soil aggregating protein, it will improve soil aggregating properties of the soils. So this is the diagram will say how it will be useful. As I already told, this mycorrhiza, this is kind of root, this will move just away from this root, few centimeters away from this root. Whatever nutrients available from this away from the root, slowly it will mobilize and available to the plant systems. And also it's play a major role in this water holding capacity. As I already told, it's producing kind of peculiar kind of protein that is the globally soil aggregating protein. This protein play a major role for soil aggregation, through which we can improve soil aggregation. So by which we can enhance this water holding capacity of the soils. Apart from this, these microorganisms also trigger some metabolic pathway in our plant system. It will induce or improve immunity systems in our plants. It's a kind of additional root hairs, so by which it will provide more surface area in these plants. It will take more nutrients and water. So by which it will improve the growth, water management, drought management, and overall it will improve the plant health and increase the yield in many agricultural crops. As a biotic stress, biotic stress means maybe the disease and these are some pathways. By producing some, triggering some pathway like salicylic acid pathway, phenylpropanide pathway, or ethylene pathway, jasmonic acid pathway. By triggering some pathway, these organisms will produce some phenol, improve these lignin contents in this plant, and produce some antifungal compound, oxidative enzyme, some low molecular weight compounds, through which it will bring this resistance or manage disease or nematode in most of the agricultural crops. Two of the students have successfully done some work on this mycorrhizal helper bacteria. Similar to PGPR bacteria, this mycorrhizal spores is being associated with some beneficial bacteria. When we apply this mycorrhizal helper bacteria along with this mycorrhiza, we can get better response. So these things already we have proved and also published in many papers. These mycorrhizal spores being associated with a lot of actinomyces, that is actinobacteria, streptomyces, and plant growth promoting bacteria. Those bacteria also have tremendous roles in this disease management and nutrient solubilization and plant growth promoting activities. Here you can see some of the pseudomonas being associated with these spores has good biocontrol activities. This is some of the streptomyces species is associated with these spores, effectively managing or suppressing this Fusarium alternaria. Also, we have proved that these organisms also have this phosphorus solubilization, zinc solubilization. Apart from this, they have good lignocellulitic activities. Already our students have proved that by using this mycorrhiza along with this mycorrhiza helper bacteria along with this mycorrhiza, we can manage rhizobium, this banana wilt management, banana wilt caused by this Fusarium by using this streptomyces along with this mycorrhiza. As I already told, this mycorrhiza producing lot of protein, soil aggregating protein, that is specially called glomalin. This glomalin play a major role in soil aggregation. Those things also we have proved in many horticultural crops. Here you can see when we use this mycorrhiza alone, we could record around 10 milligrams per gram of soils in the rhizosphere. When we use this mycorrhiza along with this mycorrhiza helper bacteria, at least 20 to 25% we can increase. That means if we use mycorrhiza helper bacteria along with this mycorrhiza, we can bring more changes in our agricultural crops. Yeah, this is another technology in continuations of ARCA fermented cocoa pit. As I already told, the people are using this cocoa pit or the decomposed or fermented cocoa pit for their seedling productions. Here we thought that this mycorrhiza has tremendous role, particularly in horticultural crops, plantation crops, as well as other agricultural crops. But we have to apply large inoculum. For example, as per substrate-based inoculum, if we want to apply, we have to apply nearly 100 grams per square meters. So that means per hectare, we have to apply huge amount. Where applying the huge amount is very difficult for farming communities. That's why we have developed one simple technology. By using this ARCA fermented cocoa pit as a substrate, whether is it any possibility to produce ARCA arbuscular mycorrhizal fungi. That is our idea. Here, idea is very simple. You might have heard this root organ culture, wherein their concept is they want to produce more root growth, wherein they will introduce these mycorrhiza. The same concept. Here, substrate is fermented cocoa pit, wherein we will introduce this mycorrhiza with the help of some mycorrhiza helper bacteria, through which we can produce more enough. When the people use those fermented cocoa pit along with this mycorrhiza for their seedling productions, this mycorrhiza will get into the root. It will be easily colonized. When it goes to the field, they need not apply again this mycorrhizal fungi under field conditions. So this is the overall diagram how we can produce the soilless mycorrhizal inoculum. This is like fermented cocoa pit, wherein we have to apply these mother cultures, introduce this microbial consortium, mycorrhizal helper bacteria. Then if we raise some host plant, after 45 days, we can produce the huge inoculum. These huge inoculums containing alpha fermented cocoa pit, if you blend with normal fermented cocoa pit, then this will act as an excellent biological source for raising crop plants. As per Bureau of Indian Standard, we have to maintain 100 spores per gram of soil. When we use this technology, even we could achieve more than 10,000 spores per gram materials. So, yeah, another important area. Now people are giving special attention to endophytic beneficial microbes in our agricultural systems. Endophytic means when we introduce any microbes into the soil, that microbes have to compete with the naturally occurring microbes, because soil itself contains a lot of other microflora. Sometimes the competing with the natural microflora sometimes will be difficult. That's why in most of the cases we used to get some failures in this microbial inoculum. That's why to avoid those stress, the people are giving special attention to endophytic organisms. This is again my colleague has developed this endophytic inoculum exclusively for rice. Because we might have studied Actinobacteria is a free living organism, will survive only in soil. But we have proved that Actinobacteria also will survive inside this plant system. It will be act as a good endophytic nitrogen fixers. By using these endophytic nitrogen, by conducting many trials, he has proved that we can save around 20 to 25% nitrogen usage in some crop plants. And also we developed some solar, because now here I have not touched anything about this blue-green algae and other things, because it needs a separate presentation. This azolla and blue-green algae also the important nitrogenous biofertilizers being used for these rice cultivations. In azolla, the initial inoculum load is very high. That's why most of the farmers may not like this technology other than these animal feed. Because initial inoculums we may use even 500 to 1 ton. To carry the huge quantities is very difficult. That's why our colleague developed the sporocarp based formulation. Instead of applying 500 kg, even by applying this 500 to 200 to 500 grams of sporocarp based formulation is sufficient for 1 hectare field. Another important aspect, how best we can explore this microbial culture for organic waste recycling. But here I have, since I am from Rice Research Institute, now we are working on for straw residues management. Now in Punjab, Haryana is a very serious problem. The people used to burn the residues in their field. Now we are working on how best or how quickly we can decompose this rice straw by introducing these microbial cultures. Even we developed some fungal-based formulation for decomposing this rice straw under ex situ conditions. By using these decomposing cultures, so we could decompose within 45 to 50 days. Then afterwards we can use this as a good.
Paneer sir, sorry to interrupt, sir. We have another session
Also we can use for other.
So would you mind just quickly?
Already I told.
Thank you, sir.
Sikkim, we have formulated the two formulations. One consortium for nutrient management, another one consortium for pest and disease management. Both consortium were validated in multi-location trial. We got some good performance compared to conventional practices. By adopting this technology, so they have recorded nearly 10 to 15% yield improvement as compared to conventional practices. So here message is, if we use effective microorganisms or efficient microorganisms in right way, so definitely we can improve soil health, we can minimize usage of inorganic fertilizer, pesticides, we can make sustainable agricultural productions. So with this, I would like to conclude my talk. I thank our institute director, head of the divisions, for giving me this opportunity to have interactions with your people. I thank my students because they are the real workers. Because of their hard work, we could achieve. I thank personally the embassies. I thank you very much.
Thank you very much, Dr. Panneerselvam sir, for your wonderful talk on soil microorganisms and sustainable agriculture. We have few questions, and I'll just ask one by one. Can we use Actinobacteria plus for irrigated rice?
Hello.
Sir, can you hear me?
Yeah, please tell me, sir.
Sir, can we use Actinobacteria plus for irrigated rice, sir?
Yeah, we have used irrigated rice also. We have used irrigated rice, but the thing is, we have not tracked those organisms in these rhizospheres and other places. But we have used, but we not get good performance like aerobic rice. For your one more kind information, we have done some microbial diversity studies in wetland soil, wherein we have recorded very high amount of actinobacterial population. Still, we could not understand how these wetland conditions supporting this Streptomyces species or actinobacterial group in this rice soil. So that's why we have to understand some mechanisms. What is the reason behind how these organisms will perform in these waterlogged conditions?
Thank you, sir, for your nice answer. We also have a few questions that I will post you because we have another session starting at 10:15. Thank you very much for your wonderful lecture. I now invite third year student Kirtana to propose vote of thanks for the lecture.
A very pleasant morning to one and all. On behalf of our college, I feel immense pleasure to thank Dr. P. Panneerselvam, Principal Scientist, ICAR Cuttack, for giving an excellent coverage on the topic, Role of Soil Microorganism on Sustainable Agriculture. This session was very informative, sir, and thank you so much for sparing your precious time with us. Further, I feel very grateful to thank our principal, Dr. B.J. Pandian, and our professor, Dr. M. Inyakumar, for having organized this valuable session. And finally, thank you for all the participants for your involvement in this session. Thank you, sir.
Thank you. Thank you.
Thank you, Kirtana, for your vote of thanks. I once again thank Dr. Panneerselvam sir for his valuable time. He is very busy and he has spared his time. So I thank him very much. Thank you very much for all the participants for joining the webinar. Thank you. The certificate link will be sent to you to your mail IDs through your registration part later in 1 or 2 days. Thank you very much for joining.
Thanks one and all for this great opportunity, sir, to have my feeling with you people. Thanks, sir.
Thank you, sir.
Thank you, Panneerselvam. Thank you. Thank you.